You got to be careful when Dr. Newman says, I'm not going to ask you questions about this. I may have talked about it too. And I may ask a question, like somatic hypermutation, somatic recombination. He may not ask you a question about it, but I will. I guarantee you that. Okay? So you got to be careful. You guys got any questions? Have you got around to looking at this yet, or you got plenty of irons in the fire? Okay. I'll also answer emails. I'll be around Monday morning. I will not be in the exam. You will have proctors that know nothing about immunology. I don't know what to say. I haven't lectured that night. So get to me Monday morning if you have any questions. We'll get them figured out. Okay? Shall we go? All the slides that I'm going to talk to you about here, you've seen before. You have copies of it. We started off, we talked about the structures of the immune system. We talked about the flow of the lymphatics up to the lymph nodes. You have more lymph nodes in areas where there's mucosal surfaces. All the lymphocytes come from the bone marrow. The B cells maturate in the bone marrow. The T cells leave the bone marrow, go to the thymus, maturate in the thymus. We have the spleen, which is basically a lymph node for the blood, sucking those bacteria out of there. Everything flows up the lymphatics, dumps into subclavian veins, goes around in the blood. Everything circulates. Comes back, oozes out into the tissue. We have two branches. The innate, which is non-specific. It'll hit bug A, bug B, bug C, all at the same strength. It's non-specific. Its response is sort of fixed, it's limited, and it's constant, as opposed to the adaptive response that is highly specific and will improve with exposure. We have the cells here. We really didn't talk about the cells over on the right, the erythroid lines. But here we got the monocytes, macrophage, dendritic cell, mast cell, granulocytes, neutrophils, basophils, etc. And then we've got the lymphoid progenitors over there that come up with plasma cells, B cells, T cells, and your NK cells. Okay? And that becomes more important in some of Dr. Newman's future lectures when he started talking about various lymphomas. We talked about the initial defense, the skin being a nice tight epithelial border. We have the mucous membranes, which are a perfect target for invasion of microorganisms because those sites are where we exchange fluids, gases, etc. So we do not have that tight boundary. Bugs try to get in there, they do very nicely. We talked about the components of the innate system, both mechanical, chemical, and of course we've got normal flora on all sites. We talked about the basic process, trauma, introduction of bacteria into the cell, damage of the cell, produces cytokines, bacteria run into macrophages, hanging out in the tissue. Macrophages are mature monocytes. Monocytes circulate throughout the body and the blood. They find some place to go. They go into the tissue, hang out there. They're macrophages. The macrophages eat the bacteria. They produce more cytokines. All the cytokines induce the inflammatory response. We have the hallmarks of the inflammatory response. We have migration of cells into the area. Here's the neutrophils. Neutrophils are the largest number of blood or leukocytes in the blood. Their whole purpose is to come, eat, get full, and die. Once they die, they're cleaned up by the macrophages. Of course, binding of bacteria to the macrophage goes through some process of engulfment, phagosome, fusion with the lysosome. We have the NADPH oxidase there that induces the production, causes the production of various oxygen radicals that are used to kill the microorganism. Then we have the various lytic enzymes, lysozymes, etc., that chew it up, kick the bug out. Binding of the bug to the macrophage also induces transcription, NF-kappa B, etc., produce cytokines. This whole complex of the immune system, the immune response, is regulated by which cytokine is produced in response to what action. Dr. Newman talked about that. If innate immunity doesn't get the job done, we start going into the adaptive immunity where the dendritic cells grab the antigen, grab the microorganism, travel through the lymphatics to the lymph node. They get down to the lymph node. They present antigen to naive B T cells. Look for something to stimulate. If you stimulate something, then you get clonal propagation of that cell type. Circulation of the lymphatics right there. We try to keep things from circulating in the blood supply, but bugs get around that. We have a primary response, which is IgM. It takes a while to get there, 10 to 12 days, something like that. If you see that antigen again, you get a much quicker, much stronger response from memory cells. Okay? But that memory response is only specific to that given antigen, not another bacteria. Okay. We have a variety of innate responses on various areas, intracellular, extracellular, surface, etc. We have the complement system. Complement system bridges the innate immunity and the adaptive immunity. The first 2.5 processes of the complement system, the alternative pathway, which is stimulated by simply binding microorganisms. You get C3b going. You get lectin binding from the mannose binding proteins. These are the acute phase proteins. You get that going. You get either C-reactive protein or antibody complexes to get the classical pathway going through C1. I don't care which one starts the process. In the end, they all do the same three things. They recruit inflammatory cells. They act as opsins to coat bacteria, facilitate the uptake, and they poke holes in membranes. All of those lead to death of the pathogen. Here's C3b, leads to opsonization, specific receptors on the macrophage, enhanced phagocytosis. Here's C5b, poking holes in the membrane, the MAC complex, membrane attack complex. The little parts of the C3, C5, C4 that are produced, those are amphalotoxins. They go off, they increase vascular permeability. That gets the bugs to the, or the blood cells to the area, leukocytes to the right area. Now, on the macrophages, there's all kinds of receptors. There's toll-like receptors, there's LPS receptors. Once you bind these receptors, it's going to trigger some type of cascade, some kind of cytokine production. And here we have all the cytokines, IL-6, fever, TNF-alpha, goes through a couple things, gets to fever 2. IL-1 beta ends up with fever. You've got CXCL1 or CXCL8 or IL-8. That's chemotactic factor. And then we have IL-12 that activates NK cells. And what did I just do? Hit the wrong thing? Yeah, I did. Hit the wrong button, guys. Let me put her in reverse here. Okay, there's the cytokine. These cytokines, the first three, the pro-inflammatory cytokines, they go on to make the acute phase proteins. They suck neutrophils out of the bone marrow. They stimulate the hypothalamus to increase body temperature, and you get breakdown of various metabolic substances to fuel this entire process. IL-6, here you produce C-reactive protein, mannose-binding lectin. Those are going to go out, bind bugs, get going with the complement system. Here's the complement system. Nice cascade. C3, the ones you really need to know is the C1, C4, C3, C5. Yeah, those are a lot. There's all these other things. I understand that. But there's over 50 proteins involved in this. We're just touching the surface of it. Innate viral protection. Boy, do I have some of that going on this morning. Woke up last night, felt this little thing going on in the back of my sinuses. Somebody broke rule number 1. Somebody sneezed at me. Well, wait a second. My wife gave me a kiss last week when she had a cold. Close. Yeah, just bless her heart. You know what's coming.